Good morning. In the last video, we talked about probability, and we said that probability is the likelihood of occurrence of a particular event, or in other words, how possible the occurrence of an event is. And we said that there are two extremes of probability. The first extreme is probability zero, and probability zero means that an event cannot happen at all. The other extreme is probability one. And when we say probability one, we mean 100%. So if you say that the probability of an event is 100%, this simply means that there is 100% certainty that this event will occur. And probability between 0 and 1 is in between probability 0 and probability 100%. For example, if I say that the probability of a particular event, for example, the visit of somebody or the occurrence of a natural phenomenon is 0.3 or 0.5, 0.3 means 30%. So there is a 30% probability that this particular event can occur. And the most important type of probability that we use in genetics is a famous example that we always give about the probability of having boys and girls in the offspring. For example, if I say that if this is, for example, the chromosomal pattern of the mom, so if this is a maternal chromosome, for example, M, and the mom has two loci for the color of the eye, and suppose that these two loci are occupied by two dominant alleles. And we said before that the allele is the alternative form of the gene, so she has the allele that is dominant and that is responsible for the brown color of the eye. Whereas, for example, the dad has, I'll take the simplest example, for example, that the dad is recessive in both loci, and he has the recessive allele for the blue color of the eyes. This is what we call the genotype of the parent. So the genotype of the mother, which is usually denoted like this. So the genotype, the female genotype, will be B capital, denoting that the two alleles are dominant. And in such a case, we say that the mom is homozygous for the dominant trait. And homozygous for the dominant trait or the dominant characteristic means that she has two dominant alleles. Similarly, the mom is homozygous dominant, and the dad is homozygous again, but recessive. Why? Because the genotype of the dad is B small, B small. So this line in your genetic diagram represents what we call the genotype, or in other words, the genetic makeup of the individual. This is the genotype of the parents, and if we come genotype of the parents, which we denote by a P. I'm sorry, it's not denoted by a G, it is denoted by a P. And here, when I write P to the left, I mean that I have the parental genotype P. And then there is another line, which is denoted G, which is the possible gametes. So during meiosis, you finally get 4 daughter cells, but we represent them only by 2 in your genetic diagram. So we have 2 possible gametes here, and I'll be explaining later on when we take the details of the genetic diagrams why we don't write the 4 possibilities of the 4 gametes. And so we just write the 2 possibilities. So it is either B capital or B capital. And since they are similar, we sometimes find that it's enough to write only one of them. Similarly, here we have one possibility for the gametes. So what we wrote here is again a genotype, but here it was the parental genotype, meaning that we were writing the genotype of the somatic cells, the original cells, but here we are writing or representing the gamete genotypes. So all their offspring will be B capital, B small. There is no other possibility. Since brown is dominant, so if the genotype, this is the genotype of F, which is the first filial generation or the offspring, then they will be heterozygous or hybrid because they are made of one dominant allele and one recessive allele. This is the offspring genotype of their somatic cells, but their phenotype will be brown-eyed. So all of them will be brown-eyed. In such a case, since all of the offspring will possibly have or will certainly have a brown color of the eye, then we say that the possibility is 100% or the probability equals 1. But if we come to the determination of sex in the offspring, then we will find that females in general, we know that human beings have 46 chromosomes in their somatic cells, meaning that we have 23 pairs of chromosomes. Out of the 23 pairs of chromosomes, 22 out of these pairs are called autosomes, meaning that, and all this will be mentioned in detail later on, they are called autosomes carrying properties that are not related to sex, and we have one pair of sex chromosomes. So if this pair of sex chromosomes is XX, then the individual is a female. So the genotype of a female or the sex chromosomal structure of the female is 22 pairs, of course, so it will be 44 plus XX. This is the sex chromosomal structure of a somatic cell of a female. 44 autosomes or 22 pairs of sex chromosomes. And on the other hand, we have 2 sex chromosomes, which are exactly similar. So here we have 44 autosomes. In case of a male, then we will have 44 autosomes again, but instead of XX, we will have X and Y. So the presence of the Y chromosome is crucial for the determination of the male sex. Why am I saying this? Because if you find the Y, even if besides this Y you have 2X or 3X, then these are, of course, abnormal chromosomal structures, but despite the greater number of X chromosomes, the individual will still be considered a male. So if we do a genetic diagram to determine the possibility of sex in case of the marriage between a male and a female, so the gamete, if a female sex chromosomal structure is 44 plus XX in her somatic cells, then the gametes will be haploid and will contain half the number of autosomes, which is 22, and half the number of sex chromosomes. Similarly, for the male, 44 plus XY. In the female, we have only one possibility, meaning that all the gametes of the female, all the eggs, the ova of the female will contain 22 plus X, whereas in case of the male, the sperms will be either 22 plus X or 22 plus Y. Because if you divide 44, this makes no difference, 22 or 22. But if you divide XY in the sex during meiosis, then half of the sperms of the male gametes will contain X and half will contain Y. So if we have a female and a male, then we have the female gamete 22 plus X and the male gamete 22 plus Y, or 22 plus X. We have two possibilities, either 22 plus Y or 22 plus X. So if a sperm 22 plus Y is the one that fertilizes the ovum, then the resultant will be 44 plus XY, and it will be a male. But if the sperm, we have just mentioned yesterday in the classroom, the random fertilization and how random fertilization is responsible for variation. So here we're talking about random fertilization. So here we do not know which of the two genotypes of sperms will fertilize the egg. The egg has a constant genotype. So in such a case, in the second case, we would have 44 plus XX. So the ratio is 1 to 1. So there is equal probability that the resultant offspring be a female, a male, XY, or a male. So the possibility of having a male is 50% or 0.5, and the possibility, or half. You can say that the likelihood or probability is 50%, or that the probability is 0.5, or that the probability is half. And the same applies to the female, the probability that the offspring is or be a female. If we come back, so when I say that there is a 50%, when I say in this slide, we return to this slide, and when we say that in general, 50% probability that the couple, there is a 50% probability that the couple gets a boy, and there is a 50% probability that they get a girl. This doesn't mean that in every couple you will have half of the offspring boys and half of the offspring girls, or half of them being males and half of them being females. There are families where all the kids are males and other families where all the kids are females, or other families where the ratio is 3 to 1 in favor of the males or of the females. So the presence of this probability that we have obtained from what the genetic diagram that we did, or you can do it through a Punnett square, and I'll tell you it's very similar. The Punnett square is a square where you write the genotypes of the gametes of the male and the female. For example, you write here XY and you write here XX, because these are the chromosomes that make up the difference, and then X with X, you get an XX, and then here an XX, and then here you get an XY and an XY. So half of the offspring will contain XY, and half of them will contain XX. So it is similar to a genetic diagram, but it's a way of representing the probabilities or the possibilities of genotypes and phenotypes. So a Punnett square enables you to predict the outcomes of a genetic cross, knowing the genotypes, as you can see on the Punnett square, and therefore the phenotypes. The genotype is the genetic makeup. So the genotype is the genetic makeup, like of the individual, like 44 plus XX. But the phenotype is the physical appearance of, or the trait itself. It's either phenotype, it's either a physical appearance or a trait. Here, for example, we can say that the phenotype is a female. So the Punnett square allows you to know the predicted outcomes in the form of genotypes or phenotypes. It's a representation that's more accurate than a genetic diagram, and that's useful, more useful when you need your results to be organized, especially when you have a large sample. So if the predicted outcomes do not always apply, then why are we doing this? We are doing this because we want to know if we have a large sample, what would be the outcome? So the predicted outcomes through the genetic diagrams, you can know the predicted outcomes and the predicted outcomes, you can obtain them either from a Punnett square, which is more organized and more useful in large samples, or from an ordinary genetic diagram, which would be more difficult to organize if you have a large sample. You obtain a predicted outcome. And that predicted outcome is nothing but the probabilities or the likelihood of percentage of the different offspring, probabilities. So you can get the probabilities through a genetic diagram or a Punnett square, but they do not necessarily apply when you have a small sample. Like, for example, when you have 2 kids only, you cannot prove this 50% probability of boys and girls, this 1-to-1 ratio of boys and girls. But if you consider a whole country where there are no wars, of course, to kill the males more than the females, if you have a country or if you consider this worldwide or globally, then you will find that the larger the sample, the more the outcomes or the percentage apply. Now, we have several rules for probability. We have a multiplication rule and an addition rule. Of course, probability, though very interesting, is one of the most confusing concepts in genetics and in statistics in general, but we will try to simplify it as much as we can. And the problems you have on those rules are very simple problems. So we will only explain 2 very simple rules of probability, the multiplication rule and the addition rule. The multiplication rule and the addition rule are 2 rules that help you calculate the predicted probability, But of course, the numerical value you get for the probability will not be certain. This doesn't mean that this will always occur, but it gives you the average outcome. So in general, the multiplication rule is applied whenever you have a relationship between 2 events that is and. And on the other hand, the addition rule that we'll be seeing on the next slide is when the 2 events is when the relationship between the 2 events is or. For example, I'll give you a very simple example before seeing the example on the slide. If, for example, you say that there is a probability 50% of meeting a friend accidentally, one old classmate accidentally in the street where you live. Of course, if the possibility is 50%, because, for example, this friend is living in the neighborhood, then the possibility of meeting 2 friends at the same time will be much less. So the more you restrict and this relationship, and if I say that the probability that I would meet Sarah, for example, in the street is 50% or 0.5, and the probability 0.5, for example, and the probability that I'll meet Malak in the same street is 0.5. So the probability that I can meet Sarah and Malak at the same time in the same street, of course, would be less because this is more restrictive. You are restricting the possibility because you are putting two conditions at the same time. So when we use the term and, then you are restricting the possibility. And in order to restrict the possibility, then you have to multiply the probabilities. And I'll tell you why. So the probability of meeting Sarah is 0.5, which is a half, 1 over 2. And the probability of meeting Malak is 1 over 2. So when you multiply the two probabilities, 1 over 2 multiplied by 1 over 2 gives rise to 1 over 4 because you are multiplying the numerators and denominators, and therefore, of course, a quarter is smaller than half. So if there is a relatively higher possibility of meeting Sarah, which is 50%, and an equal possibility of meeting Malak or probability, then of course the probability of meeting them accidentally in the street at the same time would be less, and it would be 25%. This must, if you want to use the AND rule or the multiplication rule, so again, the multiplication rule, although it's not written on the slides, but it is an AND relationship. So the multiplication rule applies only to two independent events. What do we mean by independent events? This means that the occurrence of the first event does not nullish or annul or change the possibility of the second event, meaning that if I meet Sarah in the street, this does not imply that it is impossible to meet Malak. So meeting Sarah does not mean that I cannot meet Malak accidentally in the street. And this is what we call two independent events. The occurrence of the first event, or the probability of occurrence of the first event, however big it is, like 90%, for example, does not affect at all the possibility of meeting or of occurrence of the second event. For example, in the case of sex determination, the chance of a couple having two boys, first pregnancy gives one boy, second pregnancy gives another boy. It depends on two independent events, because when the mother is pregnant with a male fetus in the first pregnancy, this will not affect at all the probability of the sex in the second pregnancy, because there is no relationship between the first process of fertilization and the second process of fertilization. So they are two independent events. What is the probability of the first child being a boy? As we say from the genetic diagram, as we said before, the probability in general of getting a boy in any pregnancy, in any fertilization, is 50%. So there is a 50% probability of getting a boy in the first pregnancy. Similarly, there is the same probability, 50% or half, in the second pregnancy. So the chance that the couple will have a boy and another boy in the other pregnancy, so it's not a boy or a boy, it's a boy in the first pregnancy and a boy in a following pregnancy. Not necessarily, by the way, that they are sequential. So a boy and another boy. So whenever I say and, then I am restricting, because I'm putting two conditions instead of one. And if I want to restrict, if I want to use the and relationship, then I use the multiplication rule. So the possibility of having a boy in first pregnancy is half. The possibility of having a boy in the second pregnancy is a half. And of course, the probability of having a boy, then a boy, is 1/4, smaller probability. If I want to restrict more and say I'm putting 3 conditions instead of 2 conditions, the probability that in 3 pregnancies, whether they are sequential or not, the probability of getting a boy in one pregnancy is a half, In the other pregnancy, it is another half in the third pregnancy. So the probability of getting 3 boys, not necessarily in sequence, then it would be half multiplied by half, multiplied by half. It would be, of course, smaller. So the bigger the number of events and the more the conditions you put, and you want all these conditions to occur. When I say and, I want all these conditions to occur. And so remember that and is restrictive. Why is it restrictive? Because it's less flexible. It's less flexible. And why is it? These are informal terms that I'm using to make you remember this multiplication rule. It's restrictive. It's less flexible. It decreases the probability because you want everything to occur. You want all things to occur. You want a boy, then a boy, then a boy. You don't say a boy or a girl. It's a boy, and then another condition. I'm very strict. I want another boy, or I'm expecting another boy, and then a third boy. This is the multiplication rule. For the addition rule is used whenever we are less restrictive, we are more flexible. We are more flexible. Like, for example, if I ask you over the phone, would you like me to buy you chocolate donuts or a strawberry donuts? So you say anything, chocolate or strawberry. This doesn't make a difference. So this is or. So or you are more flexible. You can accept this one or that one. So in such a case, you are enlarging the probability. You are increasing the probability. And in the addition rule, so we are adding, we are not multiplying, because if I have probability half for getting a boy, for example, and a probability half for getting a girl, so of course I cannot say half for another boy because I'm going to use or, so I can say a boy or a boy. So if the probability of getting one boy is a half and the probability of getting a girl is a half, so the probability of getting a boy or a girl, this or that, then you are expanding the possibility. You're becoming more flexible. So in such a case, we're adding, so the probability will be a half plus a half, which is 1. So see how the addition rule increases the probability and the addition rule expands the probability just because you're more flexible and you're using the or relationship. So the addition, of course, here the events are mutually exclusive. What do I mean by mutually exclusive? In the previous slide, meeting Sarah did not abolish meeting or cancel or nullify meeting Malak. So the two events were independent. But here, having a boy, unless of course they are twins, having a boy will prevent the couple having a girl. So you cannot have both at the same time. I'm talking, of course, about regular kids, regular newborns. I'm not talking about twins. So here we can say that the two events are mutually exclusive, meaning that if one of them occurs, the other cannot occur. So they are rather dependent events, although there is a very slight difference between dependent and mutually exclusive. They are not exactly the same thing, but for the moment being, you can say that they are mutually exclusive. Having a boy means that you cannot have a girl. In addition to this, the addition rule is used not only for or relationships and mutually exclusive events, but it can be used when more than one arrangement. So here, of course, I can see that the slide is not well written because we took only one very specific example, which is related to the arrangement of the events. So you can have a boy, then a girl, or you can start by a girl, then a boy. So this is the first arrangement. That is the second arrangement. And of course, you cannot have both arrangements in the same pregnancy and in the same childbirth. So there would, of course, be the relationship or. So the way the slide has been written, as it was written, of course, in the original reference, gives you the impression that the addition rule is only used whenever we have two possible arrangements, although it's more, it can be more expanded than this. But for the moment being, we are going to explain the example present on the slides. But anyways, whether we are considering the examples I've just explained or the one on the slide, we are always talking about the or relationship. So the probability of two different arrangements, like here, will be the probability that the first event occurs plus the probability that the other event occurs. Plus is an expansive relationship or function because we are more flexible and we are increasing the probability. So if we talk about a specific arrangement, the probability of having the girl first and then the boy would be half multiplied by half. So it would be 1/4. Why did we multiply? Because we have a girl, the possibility of having a girl, and then the possibility of having another event, which is the boy. And if we want to say that we had a girl first and then a boy, yet the relation is and in the first statement. So don't be confused. We will take every statement alone. In the first statement, I had a girl and then a boy. So I had both, but in two separate childbirths. So I am restricting the probability, a girl and then a boy. The probability of the first is a half. The probability of the second is a half. So the probability of having both together, all of them, both together, is multiplication, is less, because when we say both, we are putting more conditions. I need a boy and then a girl. Similarly, in the second pregnancy, or in the second probability, having a boy, then a girl, in this probability, you still had the two. You had a boy and a girl, so you had and, again, and, but this time it's the order that is different. So again, because both kids arrived to life, then they are there. Then the probability of having them together in our life over 2 years, over 10 years, whatever the duration is 1/4. Why is it restrictive? Because we had two conditions, a boy and then a girl. But here we have two probabilities. So starting from these two pieces of information, if we want to calculate the probability in general of having a boy and a girl, it would be 1/2. Why? Because here the probability, we have first probability or second probability. So it's either the first order, it's either the first arrangement or the second arrangement. And it is here where we used the or. So to summarize or to wrap up this slide, then we can say, for example, that having a boy and a girl in that arrangement has the possibility of, or the probability of 1/4, because we want the two conditions to occur, so we are restricting. The possibility or the probability of having a girl and a boy, the order here makes no difference for each statement separately, and a girl is again 1/4. I repeat that the probability makes no difference, the order makes no difference for each statement separately. But if we say, if we want to talk about the probability of having a boy or a girl in general, then we are becoming more flexible again, so we are expanding the probability. So it would be an addition rule, because or here means that we want this or that. The order does not make a difference, and therefore we want to expand, and we are using the addition. As we said before, Mendel is considered the father of genetics. Mendel is the first heredity scientist that introduced math to genetics, although he didn't know anything about the structure of the nucleus, of the chromosomes, of the alleles. He called them factors, but he introduced math. He talked about the probabilities, and that's why, furthermore, Mendel worked on the pea plant, which is cheap, which produces a large amount of offspring, and therefore the large sample number allowed the results of Mendel's experiments to be more or less accurate. Mendel had different laws for genetics, and Mendel's first law says that when two organisms are homozygous or pure for two opposing traits and they are crossed, then this is very similar to the example I've just explained about the color of the eye. Then they will all be hybrid, having two different alleles, and their phenotype will only express the dominant trait. He didn't talk about alleles, he said trait. So here we have the parent. This is, of course, this is not a Punnett square. This is a genetic diagram. There is a line for the parent's genotype, another line for the parent's phenotype, a line for the gamete genotype, and notice that the gamete genotype is always haploid, that meaning that you write only one letter, and then the offspring, and then the phenotype of the offspring. So here we have the parental genotype, the parental genotype. This is the genotype of the parents. And then here we have the phenotype of the parents. And then again, we have the genotype, but the genotype of the gametes. And then we have genotype, but of the offspring. And then we have the phenotype or the trait of the offspring. So we can say that this is genotype P of the parents. This line is for the phenotype of the parents. And then this line is for the genotype of the gametes, and then the genotypes of the offspring, and then the phenotype of the offspring. So when you start by two homozygous individuals, and homozygous means that they have two similar alleles, either two dominant alleles or two recessive alleles, and then you do the genetic diagram, and we usually encircle the gametes, then their offspring will all be hybrid. So we have 100%. The probability of having heterozygous or hybrid individuals is 100%. Heterozygous means that they have two different alleles, one dominant and one recessive, but why do they all show a similar appearance or a similar phenotype? This is because the dominant allele usually masks the recessive allele by definition on the phenotype. So they all appear tall, although inside their genes they have this recessive allele for one simple reason, that T capital, or the allele for tall, will mask the allele for short. And therefore, the phenotype that appears on the individual is always that of the dominant trait if present. If you want to write the same thing in the form of a Punnett square, you would write it like this. The gametes of the genotype of the gametes of one parent here, the genotype of the gametes of the other parent here, and then the results of the sum of this allele plus that allele, and then this allele plus that allele, and we go in order, this one with that one, and then this one with that one. It's so simple. The other law that Mendel talked about is the law of segregation. You all know that during, if you have, for example, during metaphase, you have this homologous pair of chromosomes along the equator. You all know that there will be separation of the chromosomes, of whole chromosomes in anaphase of meiosis 1. And then you also know that in meiosis 2, you don't have a pair of chromosomes, but the chromosomes are arranged single file on the equator, just like in mitosis, but the difference is that here we have recombinant chromosomes that have alleles from the dad and from the mom. So if I have T capital here and T small here, then during anaphase and then during telophase, during anaphase 2, and then during telophase 2, can say telophase or telophase 2, then there will be separation. So the T capital will be present in one gamete, and the T small will be present in the other gamete. And this is what we call the law of segregation or the law of separation. And we cannot see this segregation unless we are carrying out meiosis. So during the formation of gametes, the two traits carried by each parent separate. Why didn't Mendel say the two alleles? Because Mendel didn't know anything about the molecular composition of the nucleus or of the genes. So here we have separation of the traits that are carried by the parent so that each trait goes to one gamete. And the best experiment that Mendel did and that proved this law of segregation was what we called the monohybrid cross. So the monohybrid cross is a genetic test or experiment that was done by Mendel, and through that experiment, Mendel could deduce the law of segregation. So it's not the law of segregation that was set first, and then Mendel did the experiment of a monohybrid cross. No, it's the reverse. He did the monohybrid cross, and through the monohybrid cross, he understood that there was segregation. What do we mean by monohybrid cross? In such a case, the term is a bit confusing because mono means one, and it gives you the impression that there might be one parent. No, there are two parents as usual, but monohybrid cross means that we are focusing on one trait only during the experiment, because when Mendel is doing the experiment or was doing the experiment on the pea plants have several characteristics, and he studied 7 of these characteristics, but in every experiment, he was focusing only on one trait, for example, or on 2 traits maximally. So in monohybrid cross, he was focusing only on one trait. So in this experiment, for example, if I'm focusing on the height of the plant, then this is one characteristic, so it is a monohybrid cross. If I am focusing on 2 traits, I am studying 2 traits, the height and the color of the flower, for example, it would no more be a monohybrid cross, it would be a dihybrid cross. So here we have a monohybrid cross. So and hybrid means that the 2 alleles are not similar. One is dominant and the other is recessive. So if I start by 2 individuals, each being hybrid for one characteristic only, then the probability of the offspring would be 3-1, or would be 75 tall to 25% short, or 75% dominant to 25% recessive. And if we analyze more the tall ones, we will find that 1 out of the 3 is pure and 2 are hybrid. And we can see this on the genetic diagram. What's important about this is that in the monohybrid cross, there was a striking piece of information that led Mendel to deduce the law of segregation. What was this striking piece of information? That I have a characteristic which is short, being short, that was not showing on the parents, because notice that this is just a genetic experiment. Mendel did not open the cell. Mendel did not open the nucleus and see what type of genes were present. He was just observing the plants from outside. So he found out that although he crossed 2 tall plants, yet there was a trait or a characteristic that was not present in the parental generation, and that's now present in the filial generation or in the offspring. And this made Mendel deduce this way of separation of the alleles that he showed on his genetic diagrams showing segregation. So here, this is a monohybrid cross. Of course, we are using a Punnett square, and the diagram above is a genetic diagram. And you can see that if you start by T capital T small for one parent and T capital T small for the other parent, then the resultant gametes will be like this. We have 25% or 1/4 that are homozygous tall or pure tall, 25% that are homozygous short, and 50% that are tall but heterozygous or hybrid. It doesn't matter whether they are homozygous. It wasn't at the time. It didn't matter whether they were hybrid or pure, because for Mendel, all of the 3 were tall plants. So he could only deduce that there were 3 tall plants and 1 short plant, or 3/4. We can say 3/4 that are tall and 1/4 that is short. And this raised a very important question, which is, how did this trait appear? And this is what, of course, that was a genius, a very smart conclusion from the part of Mendel, that the regulation of a trait, that the regulation of a particular characteristic or trait is not done is not achieved via only one locus on the chromosome, but via 2 loci with 2 possibilities, with 2 alleles, an allele in this locus, an allele in the other locus. The 2 alleles are different. So when you have tall plants or dominant characteristic plants that give you in the first generation recessive phenotypes, this means that the recessive was hidden and it reappeared in the second generation. And this led Mendel not only to the law of segregation, but to the law of dominance, and how the dominant trait masks the recessive trait. This also led Mendel to a very important experiment, which is test crossing. When you have a dominant phenotype, like, for example, a tall plant or whatever dominant phenotype, you can never know whether this tall plant is pure, T capital, or T capital, T small, because you can make sure only when you see the nucleus, when you see the chromosomes, and not only when you see the chromosomes, when you have enough technology to decipher the DNA and know whether this is the sequence for a dominant allele or a recessive allele. But through a simple genetic experiment, Mendel could know this. So the main importance of the test cross is that if you have a dominant phenotype, meaning that the trait tells you that, for example, the plant is tall, then it has a dominant phenotype. But you want to know whether it's really containing, or it's only containing dominant alleles, or containing one dominant allele and one recessive allele. Like here, for example, having brown eyes, we cannot, of course, do a test cross on human beings, but suppose that we are discussing brown eyes, for example. So if you are pure for dominant eyes, then you cannot do a test cross, but you can deduce from your offspring. So if you are purely brown eyes, you can also be a hybrid. You can never know if you have brown eyes, but you can know either from your parents, or either from the genotypes of your parents, or from the genotypes of your offspring. So if you get married to a blue-eyed individual and you get all the offspring brown-eyed, 100% brown-eyed, then you're definitely dominant. But if you get you get married again to a recessive individual and 50% of your kids are brown-eyed while 50% are blue-eyed, then you are definitely hybrid. The other law is the law of independent assortment. We talked first about the law of segregation of gametes, and the law of segregation of gametes was deduced from the monohybrid cross. Again, this is the relationship between a monohybrid cross and the law of independent assortment. So I emphasize that it is the monohybrid cross that led Mendel to deduce the law of segregation of the gametes. And then we talked about the back cross or the test cross, and then the law of independent assortment. How did Mendel understand the law of independent assortment? Through another test that is called a dihybrid cross. So again, we have two crosses, a monohybrid cross and a dihybrid cross. The monohybrid cross led Mendel to the law of segregation, and the dihybrid cross led Mendel to the law of independent assortment. What about the test cross? The test cross just helps you know whether they are they are pure homozygous or a hybrid. So in this case, Mendel this time did a cross, but a dihybrid cross means that he worked on two individuals that were apparently dominant for two traits. This time it's a dihybrid cross. So I'm no more studying one individual, but two individuals. So the dihybrid cross applies when you are testing two individuals, and those two individuals are hybrid for at least two traits. And these traits are not on the same chromosome. For example, here we have tall plants, T capital, T small. So the plant is hybrid for the first characteristic, which is the height, and then having yellow seeds. So there is a Y capital for yellow and a Y small for the recessive characteristic, which is a green seed. So when Mendel carried out this experiment, he was no more focusing on one characteristic only, but he was focusing on two characteristics. And that's why it is called di, not mono. Why do we say hybrid? Because each of the two plants was hybrid for each of the two selected characteristics, of the two characteristics on which Mendel focused. How did Mendel know that they were hybrid? He hadn't yet known the offspring from the parents, because they were not pure. If you want to get a pure plant for your experiment, then all the parents, all the generations above or before this plant should be homozygous and pure. But he selected plants that he knew were coming from a dominant parent and a recessive parent. So he was almost sure that they were hybrid. What did he obtain? He obtained, but as I said before, the only condition, or It wasn't a condition at the time, honestly, but this was an accidental finding that helped Mendel, because we always say that Mendel was lucky that his results were correct and stayed correct for years and years before the discovery of characteristics that did not obey his rules and that were called non-Mendelian characteristics. Why was he lucky? Because accidentally, all the characteristics that he worked on, like the height of the plant and the color of the seeds, were present on different chromosomes. Present on different chromosomes, meaning that they were non-linked. It was an accidental finding. It was good luck for Mendel, but later on, years and years after Mendel, when scientists worked more, they found that there were some characteristics, for example, characteristic A and B, that were present on the same chromosome and that breached all the results of Mendel. This doesn't mean that Mendel's results were not correct. So they categorized the results into Mendelian characteristics and that obey Mendel's rules and non-Mendelian characteristics. So how did independent assortment, how did the dihybrid cross prove the independent assortment? Let me first show you the Punnett square for the independent assortment. So if I have a plant that is hybrid for both, so T capital, T small, and then Y capital, Y small, hybrid for the height and hybrid for the yellow seeds, then in such a case, there are 4 possibilities for the gametes. So you can take first with first. One gamete contains T capital, Y capital. One gamete will contain first with second, T capital, Y small. One gamete will contain second with first, T small, Y capital, and one gamete would contain T small and Y small. So there are 4 possibilities of gametes for the first parent, for the first plant, and the same possibilities for the second plant. If you look at the results, you would find that there are 9 possibilities that are dominant for both. See here, dominant for both, dominant for both. Dominant for both, I'm talking about the phenotype. So here, this is tall and yellow. Tall and yellow, although hybrid yellow, we are considering the phenotype. Tall, yellow, 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 and that's it. So if you count the number of individuals that are tall, yellow, you will find that there are 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have 9 tall yellow individuals. And on the other hand, we have only 1 short. Here we say that it is dominant for both, tall, yellow, and then recessive for both, short, green. It's only 1. And then you have tall, green, and you also have short, yellow. 1 is dominant, the other is recessive. 1 is recessive and the other is dominant, whose probability or percentage is 3. So the ratio finally of a dihybrid cross is 9 to 3 to 3 to 1. 9 dominant for both, 1 recessive for both, 3 dominant for one and recessive for the other, 3 recessive for one and dominant for the other. So this is the result of a dihybrid cross, and the phenotype ratio, of course, is 9 to 3 to 3 to 1. So the probability of having, of course, the total number of offspring is 16, as you can see, because we have a table where we have 4 gametes horizontally and 4 gametes vertically, and therefore 4 by 4 gives 16 possibilities. So if I say tall yellow, dominant, dominant, this is 9, 9 out of 16. If I say recessive, recessive, short green, this is 1 out of 16. And then we have 3 over 16 and 3 over 16. This is another form how we can write the results of a dihybrid cross. How dihybrid cross is related to independent assortment of the chromosomes. Independent assortment of chromosomes means that there is a different way that chromosomes adopt to arrange along the equator. For example, if you have this is the plant being hybrid for the first characteristic and hybrid for the second characteristic. So we have a homologous pair of chromosomes here and another homologous pair. Here the equator is horizontal, so they can arrange like this. You have the paternal chromosomes are the blue ones, for example, the maternal chromosomes are the pink ones. So you can have A capital, for example, we will say that this is up and this is down. So you can have dominant and dominant up, recessive, recessive down, or you can have dominant and recessive up and recessive and dominant down. So this depends upon how, if this is, for example, if this is the equator of the cell, so would they arrange like this, paternal, paternal, maternal, maternal, or would they arrange like this, paternal, maternal, and then on the other hand, we have, for example, maternal, paternal. So this is what we call independent assortment of chromosomes, or the explanation is how these factors are distributed around the equator, how they segregate, or I wouldn't say segregate, I would say assort, or an assort here, I mean, how would they line up? How would they arrange themselves around the equator? And of course, this is a random event. So independent assortment here means independent arrangement of the chromosomes along the equator or on each side of the equator, or independent lining up. They are why independent? We understood assortment by why are we saying independent? What allows them to be independent is the fact that they are present on different chromosomes. They are not linked. So A and B are present on different chromosomes, and this is what helped them to assort independently. So what helped them to assort independently is that they are non-linked. This is very important because if they were linked, independent assortment would be impossible. They are free to move as they wish. I think that by having said this, we would have, this is another example of independent, or it's actually the same example that I have just explained. And by having said this, then this would be the end of video 2. Thank you guys, and I'll be sending the video to you. And I hope that it was easy and understandable.